Hello. So can you hear me? Okay, good. Now, because we are going for the mixed mode, so I am here at the classroom. So this is the first time that I set up this, so it might take a little bit. So you can see my camera. Okay, good. Let's see. Okay, let's start. So you can see the PowerPoint, is it? Okay, just there's no submission button for assignment 1. I'll check the assignment setting. Yeah, for that. Yes, okay. If there's any problem, I'll fix it. Okay, just we're taking some time for setting up this computer. Now, interestingly, there's no student coming, so there's just me in the classroom. But still, we'll start. Today, we are going to talk about the synaptic transmission for the second class with more details. So we talk about the action potential coming to the synapse. Last week we talk about those synapse and all those structure. Now today we are going to go through how actually starting from the action potential arriving at the synapse and how the vesicle actually released it, and then what's the whole process being released and then activated in the postsynaptic terminal, and then being inactivated. Okay, so first you can see that there's a here a picture. The first step, the transmitter is being synthesized and then stored at the vesicle. Okay, we will talk about those, how that is being happen as well. Now, when the action potential arrived at the postsynaptic terminal, that will trigger the depolarizations of the voltage-gated channels. So here, the calcium channel, sorry, voltage-gated calcium channel. Now, if you still remember, this is one of the actually important channels we introduced it in the voltage gauge channels. apart from the one that we talk about, the sodium and also the potassium, this is also important, the calcium, voltage-gated calcium channels, okay? So that will trigger opens, the calcium will rush in, and then that will trigger the, now they started the email face-to-face. If you want to come in, I'll be here, so. But you need to check your temperature and so on, just follow the guideline. The release of the neurotransmitter, the calcium trigger, the release of neurotransmitter, and then the neurotransmitter will release and then diffuse through the synaptic cliff, okay? And then that will bind to the receptors, depending on the receptor. We will talk about the receptors in the next, starting from the next sections. Now that will depends on which type of receptor. Some of those are ligand-gated ligand-gated channels, and then the ions will rush in, and then trigger the formation of the changes in the membrane potential. Okay. Now those will diffuse out, and then also will be the neurotransmitter will metabolize it or uptake it, depends on which type. Okay, depending on the type of the system in which that we will go through those one by one. Okay. Just a little bit, it's a bit hot here. So let me just hold on a second. Okay, so let's continue. And those are diffused away. And then and there will be a process of endocytosis. Somebody asked, your classmate asked, where is the classroom? This is 4619. You should get the information, I guess. I don't know. The school or the university should release those information. So you should check if you want to come to the class. Okay, the face-to-face. Okay, here is the endocytosis to capture those membrane, and we'll talk about those as well. And this is just a very brief summary. We'll go through the process one by one. I don't know. This is actually I got the email, and I think the university announced that today is the start of some of the class will face-to-face. Some of the one will still be online totally. So for this class, it's a mixed mode starting from today. Okay. Okay. 4619. Okay. Yes, 4619. Yes, good. If you want to come, then you can come for the next class. It's a bit late now, huh? Okay, so there are two types of the neurotransmitters, a major type. For example, the amine and amino acid neurotransmitters. Those are small molecules in which that the enzyme at the local state. So when we talk about individual neurotransmitters, we will talk about those the production pathway. So in which emitting enzymes, different enzymes that synthesize the neurotransmitters, then that will be transported into the vesicle, okay, here, and then that will be ready for the release Now, the release is also a complex process, and we will also talk about it. So for the peptides, a little bit different. When we talk about peptides, peptides actually the precursor are synthesized in the so peptides are basically a short string of the amino acids, usually 7 or 8. It's synthesized in the ER and actually as a precursor. So when we talk about the neuropeptides, then you know that actually it's not that simple, just the synthesis and translation, but there's post-translational modifications at the Golgi apparatus and in the vesicle as well. Okay. Now this is called secretory granules, or the granules at the terminal that we talk about, the synaptic granules. Now it will, during the process of transport along the axon, then it will become mature and then down to the synaptic terminal, then that will be there ready for the release. So this is a bit different from the other one, okay? So in which that in every neurotransmitter pathway, we will actually talk about those their synthesis, their maturation, and their release and their destructions. Okay. So the release actually is by the exocytosis, but actually the exocytosis is not just a simple release of it. Actually, it depends on a complicated process, because when you think about it, they have to release at once together. And usually they are released as a discrete packet or contour. So each release is a fixed amount, and then it adds up depending on how strong is the action potential frequency. Okay. Now, image that it is the synaptic vesicle is stored with the neurotransmitter loaded with neurotransmitters, calcium rushing, that activate because of the action potential arrived at the voltage gated calcium channels, open up calcium rushing, and then trigger the release of the neurotransmitters. Now, after release, how they are being refilled again, that also actually involve the different possible pathway or hypothesis as well. Now, the release by the exocytosis is the calcium rush-in, and actually by special method, the calcium imaging can actually detect the real-time movement of the calcium inside to the terminal. Now, actually, those calcium are restricted into a small area, and the concentration of it can reach around 200 micromolar. So it's a regional. It's not a trigger moving up into the whole neurons. Okay, that's also a very important mechanism, because when we talk about the secondary signaling, and you know that actually the signaling mechanism is also depending on calcium. So the calcium concentration usually is actually fixed in a very localized area in the active zone that we will talk about, and then trigger the release of the neurotransmitters. Okay. So the biochemistry of exocytosis, well, actually, this is something that a lot of scientists focus on the exact mechanism of how that is happening, because that every step is carefully controlled, and then that actually, if anything happens, then that will affect the movement. And you see more and more example of how that modulation of this release or the activation of the receptors and so on can actually disrupt the transmissions of the nerve signal. Okay. Now, several steps are involved in this exocytosis. Now, for example, recruitment of the vesicle from a reserve to the releasable pool. So we'll talk about, all those vesicles, they are not randomly situated at the terminal. They have a different group. Some of those are reserved, some of those are actually in the active zone, some of those will transport to the active zone, and that actually contribute to the signal transductions. As I mentioned about, there are vesicle reserve pool, and then they will be binding to the vesicle associated protein and plasma, and approximated to the voltage gated calcium channels. So that's the reason why that they have to close to the channels, so that in a special formations or form, so that they will be ready to release once that they are there. Okay. Now this is very important because that the transmission that the synapse you want it to be fast and also to be highly responsive, and at the end, that signal, once it's done, that you want it to be stopped as soon as possible. As we mentioned about that, stopping the transmission is very important, because if it's extensively activated, then that will affect the next transmissions. Okay, that means that you're kind of freeze, So the binding of the calcium to the synaptotagment actually permits the fusion to go to completions. Okay, so we'll look at each step as exactly what happens. Actually, even for the exocytosis, it's not just the two membrane, plasma membrane and the vesicle membrane just fuse it. That's it. Because if you still remember some of the cell structure, the double lipid membrane, actually they will not fuse easily, because they actually will repel each other, because of the both are polar at the surface. So this is the cycle of it. So the synaptic vesicle are filled with a neurotransmitter by active transport here, the neurotransmitters inside, and then uptake that. And then those may be coming from the early endosome, or some coming from the vesicle that has been released and then coming back for recycle. So then they will be fused and then stored at a pool, a reservoir. And then once that they will transport to the active zones, the so-called active zone over there, and then docking. Okay. Now after it's docked at the active site, active zone, then that will be so-called priming. Okay, priming is in between. So it's a very active process in the image that it is ready to release. It just is pulling the trigger, ready to fire, just like the gun. Okay, and then there will be there's a calcium, then they will fuse it, and then release the neurotransmitters. Now after this, the process can be going back to refill with the neurotransmitters, or some will go back to the endosome. We'll discuss about why there's a so different pathway. Now, actually, the thing here is that actually we cannot see the actual process. So it is very difficult to visualize what happens exactly, because those are only the size of the vesicle can be seen by ER only, as we have some pictures to show you some of the ER pictures, and sorry, not ER pictures, electron microscope pictures. So that's the reason why that we have to depend on a lot of imaging and also biochemical study, that we know that some of this we propose how that can happen. Okay. So far, okay. This is a very interesting setting of online mixed mode class. I hope that I'll see some student coming so that I'll be less lonely. What triggers the process of priming? We'll look at the more detailed, and then you will see that what does it mean by the priming? Why there's a priming process? Now let's continue. Now, so this is the timeline. What happens? The action potential arrived, and then that triggered the influx of the calcium, and then you can see that it's in terms of milliseconds. Then the exocytosis release that. Then you'll see that the release needs to be triggered together. So that's the reason why that we people measure actually. There's oh sorry, there's some noise. Yes. Yes. Just hold on a second. You are coming for the class? Yes, come in. Oh, we have one student.
I was in the library. So.
Okay, good. So I'm less lonely now because at least I have one of your classmates. He is here. So welcome. Okay. So we'll continue with this actually. The action potential arrived, and then so it triggered the influx of the calcium, then that triggered the exocytosis. So that will be evoke the postsynaptic current. Now, postsynaptic current is a bit different. It's different from the action potential. Okay, you have to distinct about that because that is sometimes called excitatory postsynaptic current. Now that doesn't mean that when the neurotransmitter release that will trigger another action potential. If you still remember, we talk about electrical synapse and chemical synapse. The high certainty and less certainty is here. So you have to have enough trigger before that you can get your next action potential. Now, if that is enough, the next action potential will come. Okay. So you can see that those are time lag behind the vesicle will be released. Okay. Good. Well, that's what happens. Okay. So the release of the neurotransmitter by exocytosis, you can see that we are now start to talk about the component. Now, both the vesicle and the transmembrane protein are involved in that. Now, two of those are the important one called the V snare and T snare, and we will talk about those in more detail. Now, this is one of the examples how that actually combined. That is the docking, the so-called. Okay, when it's docked, this protein, particularly synaptoberrin and SNAP25, they bind together. And then there's a synaptotapman actually will detect the calcium level. Now, next to it will be the voltage-gated dependent calcium channels. You can see that actually the vesicle containing the neurotransmitters will come in, and then we'll try to dock it. Okay, and then once it's docked, and that will be in the priming situation. Now, priming means that actually is ready to release. Okay, so you'll see that there must be a group of vesicle actually is next to the active zone, and then ready for the release. Now, if you still remember when we talk about the cell structure and the synapse, actually the electron microscope pictures at the synapse, actually there's very dense dark color. That's actually where in which that those like a lot of protein or component there is forming the active zone. So here is the example. So the storage, so where the vesicle tether means that coming from the storage pool and then going close to the active zone. So they are trafficking to the target site. Now, those processes are believed to be highly organized. You can see some of the proteins here. Now, don't worry too much about those names of the protein. I'm not going to ask you to memorize those. In which the more important is how these synapse actually released the exocytosis process. So the docking of the vesicle at the active zone, and they're priming for release, and then release. Yeah, you can see that there are channels, and in which that at the very beginning they propose there may be channels, but this is not the case. Actually, we will talk about how actually that is being the mechanism, how this is actually being done. So you can see that the calcium channels are next to it. So the synapsins and the red proteins, those are some of those to mention about over here, and there is actually involved, believed to be involved in the transport from the cytoskeleton to the close to the where the vesicle needs to be. So again, every process actually is being controlled, and it's not just like floating around. So don't think that the vesicle will float around and then diffuse to the place that needs to be. Actually, they are at the pool and then transported and then linked to the active site. A lot of studies, for example, if they remove some of the protein through modern molecular approaches, they find that those vesicle transport or release will be affected. But however, actually, a lot of proteins are redundant. That means that actually it's very difficult to affect this process, which makes sense because that any possible evolutions to make it more stable will be selected, because if it is so easy to be disrupted, then the organism will not survive easily. Now, if you still remember, we talk about Botox, okay? Botox actually originates from the botulinum toxin. So actually, the toxin actually affects this, cleave this structure, so that to make the vesicle not be able to release, so stopping the transmission. So that's the reason why that in the Botox that stop the muscle from contracting, because stop the release of the vesicle. Now that's also the reason why that it is a very dangerous toxin. The reason behind for beauty treatment, actually, that's just in localized area. However, if that diffused into other area, like for example, the muscle that control respirations, then that will affect the respiration as well. Okay. So here is the docking and fusions. Okay. The interaction or distinct interactions of the proteins. So as I mentioned about, is the T snare and S snare, which is the synaptoberrin, and then the syntaxins, and then they are linked together. And in which that the synaptotapmin actually are responsible for the detections of the calcium trigger changes in the structure. Now, the whole process is basically the calcium rush in and binds with this synaptotapmin, and that trigger a structural change, and the force the diffusion between the vesicle and the plasma membrane. Okay, and then we will also have look at the possible or the proposed state or form when the vesicle at the priming state. Okay, so you like it so far? I'm just asking the one that only one face-to-face student here. So he seems to enjoy a lot.
It's been a long time since I.
Oh, he said that it's been a long time. Yes. So I guess all of you are kind of tired. Want to go back to the university. Okay. So next time, if you want to come, you can come to join. But remember, you have to take your temperature and then wear a mask. And just like me, I'm wearing mask as well, even though like it's talking like this with a mask actually is kind of difficult, but we have to do it. Okay. Okay. So far so good. All the other students. Actually, if we have more, I cannot see how many people here actually. We just have 33. Now, actually, when we have time, then maybe with more students, or if you can tell the other student, so I will release more details about the project. Group project. So because we are using Zoom, we can still do group project presentations in which that individual one can present through the Zoom, or if you are coming back to the class, we can also do that. But possibly, I will let you do it through the Zoom, unless that you have other suggestions or ideas that you want to change. Because even if you are at the classroom, you can still do the Zoom, because we want to minimize the interactions. So social distancing, even it's difficult, you still try to do that. Is it okay? So I will release the details regarding the project soon, like how many students per group, and then how that you are going to form the groups, and after that, and also the assessment method, and those details. Before it's easier because before this pandemic, when we have face-to-face, it's much easier for students. But we can still do that. Okay, I'll maybe in the next class I will do some polling, and then see how you are interested. So I will announce the next so that we can have more students here. Next class we will discuss about the project. Okay, the details, and also your opinion, because all of you are a lot of you are year 3 and year 4 students. So you have, and this is a 4,000 class, so you have more freedom to decide how that we are going forward for the project. Okay. Okay. So here is actually what happens. Now this is a so-called snare hypothesis. Now there's a so-called V snare and T snare. For example, you can see that V snare means that the one that actually at the vesicle. The T snare actually is the target membrane. Now this mechanism actually happens on all, actually believed to happen in all different targets. For example, how the other exocytosis process, like from the ER to the Golgi and so on. Now, but this one is actually specific in the synaptic terminal. Okay, there are different V snare and T snare depending on which system. Now, in here we are talking about the one that at the synaptic terminal. Okay, so those are 2 snare, that 4 step. So following the fusions to the 2 membranes, cytoplasmic membrane and the vesicle membrane, then some of the protein, for example, so-called NSF and SNAP, will bind and then disassemble the structure. Now we will talk about how that is being done. So So this is the model in which that this V snare and T snare will bind together. Okay, so it's like from the structure, it's like a tryst of the two protein. Okay, the tryst of the two protein. Now, when it is forming a very stable structure, and then twist and twist again, okay, it just like twist and twist again, that form a force at the end, and then that force the vesicle and the target membrane to fuse. Okay. Now, this structure is actually pretty stable. So you have to have protein using the ATP to release them. Okay. So that's the energy is coming from. So you can see that actually these processes will require a lot of energy. So this is only one part of it. So the synaptic transmission actually requires a lot of energy. So we'll see each part will require energy. So this is one of the part, the release of this structure, so require the energy. Now, after release, the energy is stored in the structurally. So when they form the complex, they use up the energy and then make the fusion of the vesicle to the membrane. And then you require energy to release them so that they can be used again. Okay. As I mentioned about the botulinum toxin or tendon toxin, actually affect this process. And it's like some of those toxins actually target those membrane over there. Now, as I mentioned, the vesicle is actually it's not just simply just two membrane structure. There are a lot of different proteins over there. So, for example, some of the synaptoberrin, the T snare components, and the synaptamen. Actually, that one is the one that detect the calcium concentrations. Okay. There are also different components. For example, the ATPase that actually can pump some of the ions, for example, proton ions into the vesicle. Now, you may ask why is this the case? Because we will talk about why there are pumps over there as well. And then there are transporter that transport, for example, neurotransmitter inside. And there are other components that actually facilitate the transport or interaction with the endosome as well. Now, again, I will not ask you to memorize this and ask you what proteins are performing what functions. The idea about that you know that actually the vesicle contain all those proteins actually for the different functions. And the vesicle, as I mentioned, is not just a double membrane structure containing the neurotransmitters. Okay. So far, okay. So now the active zone, actually you can see that there are different protein components, and then the vesicle will come in and then form the structure at the priming state, and ready to release. So that's the reason why that at the active zone there are different proteins over there. So one of your students asked integral synaptoberrin and synaptotamen. Yes, they are integral protein. Yes, right. Okay. Okay. Now, this is the picture that I talk about. So at the time that the vesicle are transported to the active zone, they are close by, the V snare, the synaptoberrin, okay, and the T snare syntaxin, and also the SNAP25. They will form a stable structure together. The priming transport to the active zone, and then priming to the active zone over here. Now, this is at the priming state in which that believe that they are, as I mentioned, the vesicle are loaded, and you can see that the structure may be like this actually. Half of it has been fused already, and another half is not there yet. Okay. Now, once the calcium binds to the synaptotapmin. So synaptotapmin, yes. So because it has the calcium sensing zone, and then that will trigger a release of the final part, and then the neurotransmitter will come out. Okay, so that's the reason why that this is the process. No, okay. So this is the process ready to release. Okay. Well, that's the reason why also why that we believe that the vesicle can release together at once that so fast and then trigger the activations of the postsynaptic current. Okay. So how this vesicle is being recycled, different pathways. Now, so you can see that actually there are different potential, so-called the reused kiss and run, reuse kiss and stay. All those are possible. And then, for example, this one kiss and run means that once it's in priming, and then that calcium get in, and then the neurotransmitter released after calcium. Then the membrane will go through the endocytosis, and then go back to be filled, and then uptake again. Now, kiss and stay, some of the potential is that after it's being released, it stay and then just reuptake, and then that will be going to the same process again. Okay. Now, this is difficult to determine because that we cannot see the process. This process happens really fast, and some people actually trying to image that and determine which one. And so far, it seems to be both is possible. Now, so the other part is the endocytosis of the membrane. And okay. Why is it on page 16 that it shows the natural is found on the visible side instead of the membrane? So 16. Which is 16. Yes, it's in the vesicle side. Yes. Okay, let's continue. So now endocytosis to recycle the membrane. Now, so this process actually needs to be done. So why? So one of the students asked how the vesicle are refilled in the kiss and stay mode. Now, the kiss and stay mode. So this is the thing. If you look at the pictures, so that the enzyme that synthesizes that the neurotransmitters is all over in the cytosol. Now, that means the cytosol actually contain a lot of the neurotransmitters, but the neurotransmitter in the cytosol is not as high concentration as in the vesicle. So you can still think that if the kiss and stay, they stay there, there are pump and transporter that transport the neurotransmitter into the vesicle. So still possible. Okay. So the refill part don't necessarily have to go back to the the pool or the endosome. So because the filling of the vesicle actually is by the vesicle itself, because the pump and the transporter are actually at the vesicle. Okay. Okay. Now endocytosis. So why do we need to do this process? Because that if you keep on exocytosis, then the membrane surface of the synaptic terminal will be getting bigger and bigger, the surface. And actually that can be detected as a result, So that means that the membrane needs to be recaptured. Now this process called endocytosis is going through the dynamine and caffeine. Now the caffeine actually is like a coating around the a patch of the membrane, and then actually the dynamo will come in and snap, and then that form the endocytosis vesicle again. Okay. Now it is actually very interesting. The whole process is not random, because all the vesicle size, they are pretty consistent. Okay. If you still remember the pictures that we showed, again that EM pictures, all the vesicle size, they are pretty much similar. Okay. So there's a mechanism that actually control the size of the vesicle. So that means the process is highly regulated, rather than just you patch some of the membrane and then that's it. Okay. So as I mentioned, two pathway for the vesicle membrane to be cycled after cytosis, exocytosis. So the best characterized pathway, actually, vesicle flattened into the membrane after exocytosis are recycled via the caffeine-mediated endocytosis. Okay, just like this, and then after that can, as I also mentioned, kiss and run, and then kiss and stay. Those are the ones that I mentioned already. Now the vesicle can go back to the endosome, as we talked about. Some of those are going directly back to fill up the neurotransmitters. So why we think that there are two processes? What is the advantage of these two? So you can go to endosome can go to the neurotransmitter. Endosome can go back to the vesicle.
Because you can uptake some of the molecules from the genetic cleft to modify the exon.
Well, that is actually the only class we face to just talk about. The endocytosis can uptake some of the molecules and then go back to the endosome or go back to the neurons. Yeah, that actually is true. So when we talk about the growth factors and some of the other things, the endocytosis can pick up some of the signaling molecules and go back for the signaling. Now, but this is here. I asked why there are both pathways. So this one is actually pretty straightforward. You can go back directly. This is fast, so you can uptake the neurotransmitter again, and then use it for release. So what is the other advantage of going back to the endosome? Or possible buffering effect? Yes, actually, that can be true. Actually, buffering. Now, in the sense that you can go back to the endosome, and then maybe fix some of the protein, and then some of the damaged protein can be removed, or some of the new membrane protein can be come back to here. So those we don't know, because actually it will be also very difficult for you to isolate different fractions of those vesicles or endosome to determine like what happens. But those are possible, and actually those are the things that we believe that can happen. Okay. So here is the some of the more simple way to describe it, in which the resting state, you get the vesicle, and then when this is stimulated influx of calcium, then that can be released. Okay, and then after a lot of membrane fills it, then that will be endocytosis. So this version is a very simple version, but so you can see that you can say it seems to be very simple, but actually the whole process contain different steps involved, and all of them can be controlled. Okay. Now this is what we talk about refueling. So you see that there's an ATPase pump at the vesicle protein. Now what happens is that actually the vesicle contain the proton ATPase pump. You pump the protons inside to the vesicle. So actually the vesicle kind of acidic inside. And then this proton is being used to pump the neurotransmitter feeding into the vesicle, so that the vesicle contain higher concentrations of the neurotransmitters, okay, coming from the outside. So what happens that in the synapse or cytosol, the enzyme continues to synthesize the neurotransmitters, and then it is transported into the vesicle. The energy is provided by the differences in the proton. So at once you see that, again, that I asked you, if you still remember, why that we use the pump, not the pump specific neurotransmitter. Now, if each neurotransmitter requires a specific pump, then you require different pumps add on differently. But this one can be reused. For example, this one will be similar to, for example, in the mitochondria for proton pumps, similar. So actually, in the mitochondria, you should still remember in the other biochemistry, that pumps actually can form generate the ATP, but actually those protein, it can do reverse as well. If that provides ATP without the gradient in the proton, then actually can move the proton in the opposite directions, actually, okay? So the same thing, actually. Now, transporter small molecules from the cytosol into the vesicle. Now, there are different type of transporter. Now, those in the vesicle will be those from the plasma membrane. Yes, you're right. The refueling process for amino acid and the acetylcholine, the peptides are different. As we mentioned, the peptides is produced in the ER and also mature in the Golgi apparatus and also in the vesicle. When we talk about the neuropeptides, then you will see more, okay? Yeah, good idea. So as I mentioned, the vesicle transporter and the transmembrane transporter are different, okay? They make use of different mechanisms. Here is 3 of those: the vesicle transporters, the glutamate uptake transporter, but this is actually at the membrane. And this one is the uptake of other transporters as well. Now, transporters are very important. So you have to distinguish the vesicle transporters and the membrane transporters, okay? Because the vesicle transporter actually usually use the proton pump. Sorry, not the proton. Use the proton pump and then use the proton as the transport, okay? So here is the example of the vesicle transporters. This one is the dopamine exchange for protons. So the dopamine can get into the vesicle and then concentrated at the vesicle, okay? Now, the other one is the vesicle ATPase that pump the proton inside to the vesicle. And you see that this can, concentrated, this provide the energy for the concentration of the dopamine. For the proton pump, does it exist in the beginning or appear after release of the neurotransmitter? That one, we believe that actually, so your student asks, whether the proton pump is there or before or after. Now, we believe that it's always there, okay? It cannot somehow suddenly transported to the vesicle. So that's what we believe. But there's other ways that always new findings sometime, but this is what so sometimes we think that that is the case. It is kind of difficult to prove whether it is the case or not, okay? Okay. Okay. So the neurotransmitter needs to be in activations. So we always mention about in the neurotransmitter inactivation is very important. So the reason for inactivation occurs so that the synapse can return to the functions again, fast time manner, so that it can go back to the actions. So that's the reason why that we are limited on the time of this. So that's the reason why even that the athletes, they can train and then make their response time faster and faster. But there's always a limitations, okay? And the limitation, you can see that how people can run apart from limit on the muscle power, that also limit on the nerve signaling, okay? So there's so fast that we can go, okay? Unless there's a mutant happens in the future that improve the way of how the nerve signaling can transmit it, okay? So there are three ways to inactivate the neurotransmitters: the enzyme degradations, the transport, and the diffusion as well, okay? Those are the three major ways to inactivate it. Now, in different neurotransmitters, they might be different in which way that they do. Now, let's look at it. Enzyme degradations. Actually, physiologically, only the hydrolysis of the acetylcholine is an important example in this category. So in the acetylcholine is depending on the acetylcholine esterase, is degrade the acetylcholine into choline, and then it's break down at the terminal. So and then the choline is transported back into the nerve terminal. This part is actually, when we talk about this enzyme, is actually very important in the case that actually it can be a very strong or powerful neurotoxin that can kill people instantly for a short time. I cannot remember his name. The North Korean a son of the previous. Actually, he is the half brother of the president of the North Korea. He was killed in Malaysia. You might heard about the news, by the very powerful neurotoxin, in which that toxin, Kim Jong-un's half brother, yes. That toxin is actually a powerful inhibitor for this acetylcholine esterase. Now, in which that you may ask how that killed the person, because that once that is inhibit the enzyme cannot function anymore. That means that the buildup of the acetylcholine, and then that means that the muscle will be paralyzed, and also in the central nervous system, that also affect the person, and then that person actually without the antidote, he will die like at a very fast time, and actually he was being killed within a short time. Okay, so when we talk about the enzyme, then we will talk more detail about it. So transport, this is also can be inactivation. So the reuptake from the synaptic cleft into neurons is also a major mechanism, apart from the acetylcholine actually. There are two major families of transporters are involved. For example, one take up the glutamate, okay. The other one take up the GABA, that classes. So you can see that at once the two major classes, so that a little bit modification of those can be used for the transport of different neurotransmitters, okay? Now in here, you can see that this is a glutamate transport by the sodium potassium co-transporter. So then you can see that the sodium gradient and the potassium gradient can store the energy for the transport, okay? All those transport are energy dependent. Now another example is the GABA using the chloride ion and sodium ion energy. the co-transport. Okay. Now just look at it a little bit more. So those are the transmembrane structure, and then you can see that actually in the cytosol there are more potassium and the hydroxy ion, and then that going in, and then the sodium ion will come out as well, so that to uptake the glutamate into the cytosol. Okay, so this is in the synaptic terminal, in which that the after release the glutamate that needs to go in really quick, so that to inactivate it. Okay. This is another one in which that this is the chloride ion and sodium type of a transporter, and in this case is can transport the dopamine. So you see that those two major type of transporter, if they modify, they can modify to transport different class of neurotransmitters. So at once that you will see that if you have drugs that to target the transporter, then it depends on whether it is specific or not. So if the drug targeting the transporter is not specific, then that can affect, different transporter because they are highly similar. So when you look at some of the drugs, when you do the project, then you see that actually there's some of those are quite different. Sorry, quite not selective. When we talk about the receptors, when we talk about more details on different the neurotransmitters pathway, their neurochemistry, and some of the drugs that can affect their transmissions, then you will see that actually some of the drugs are not very specific. Some of those are more specific. It depends, because some of those they are quite closely linked together. So the other inactivations is for example, diffusions. It diffuse away from the synaptic cliff. That's also another major mode of the inactivations. For example, in the peptides, that might be neuropeptides. That might be the case. Now, so you may ask, like, so why do diffuse away? Because they are attaching to the receptors. When we talk about receptors, then you know that actually the ligand binding to the receptors is not permanent. So there's an affinity. Now, if there's high concentration, that will attach to the receptor, activate the receptor. But when there's a low concentration, they will fall off again, and then they will diffuse away. Okay. So when we're going to talk about the receptors, then we will discuss more about it. So this is the final slide today. The regulations of the synaptic transmissions. Now, the synaptic transmission is a highly regulated process. So as we mentioned, so there's never an excess of neurotransmitter available to the postsynaptic receptor, except in the pathological conditions. So what do we mean? Because there's always just enough the neurotransmitter going to the receptors, and then that will be removed at a fast rate, so that to stop the transmission, so that the next transmissions can occur again. Okay, that is important, as we mentioned about. But except in pathological condition, what does it mean by pathological conditions? For example, last time we talked about epilepsy. So in some of the cases, maybe they're overactivated of the neurons and secrete a lot of the neurotransmitters, then that will be overactivated. Or in the case of some of the other conditions, for example, stroke, because of the not enough energy, and then the neurotransmitter cannot remove at a fast enough state, then that actually will affect the overactivate the receptors, and then that actually will cause the problem. So when we talk about those, then we will discuss more about those. Now, the modulation of the process. Now, the synaptic transmission can be controlled at the synthesis, release, inactivations, at the receptor level, or the integrations of all of the above. Okay. Now, that's the reason why when we talk about different neurotransmitter pathway, we always talk about the synthesis, the release, inactivation, and also the receptor control as well. So all of those can be controlled at different different level or different ways. Okay. So we'll stop here today. Any questions that you want to ask? Yes, the glial cell also help. For example, one of the examples is the glial cell can help to remove the neurotransmitters, particularly in some of the cases. For example, the glutamate transmissions and also the GABA. Yes, that's the case, and also the metabolism. What do you mean by the pull open the vesicles? Why exocytosis is hard to observe? Because the vesicle is very small. I think it's 30 nanometer. So you need to, first you need to look at, the process is also very fast, actually. Page 8. Yeah, it is very difficult to observe exocytosis. Page 8 or page. Just a moment. Page 8 first. Let's see there. Page 8. What do you want to ask? So this is page 8. Basically, is what the sequence that happens. So the sequence of the action potential arrived at, and then the time estimates that happens. For example, 0.3 milliseconds, the calcium channels open and the current rush in, and then that takes another 0.5 milliseconds to trigger the exocytosis. And then that will take time for the neurotransmitter to bind to the receptor because it diffuses. And then the time that actually, after the activator that trigger, the current, and then if that is summation of the excitatory postsynaptic current enough, then that will trigger the action potential. That's the time scale. Okay. Now, when the vesicle is stopped, that energy, the formation of the snare, actually is the structure. That energy before that, open up the structure of those T snare and the V snare. So is opening still require the ATP? Certainly, the exocytosis certainly require the ATP. The docking also required because of transport of the vesicle require the energy. The opening also require energy, but it's not directly using the ATP hydrolysis at that time. The energy is stored in the protein structure through the ATP. So it's still indirectly through the ATP. Everything is need the energy from the ATP. There's nothing that does not require the ATP. So page 16. Where is page 16? 16, yes. What about 16? Yeah, so yes, I answered you already. Okay. Okay. Any more questions? No, I think I need to let you go because you may have the next class coming soon. Okay. So I'll see you on Wednesday. If you want to come to face, you can come to face. but you can still stay online. But next week, next Wednesday, the next class, I want to discuss about the project. So hopefully, if you come to the class, more of you come to the class, online or face-to-face, and also if you have friends, so ask them to stop by or stay sometime or just come to the class so that we can have more students coming to design. The lectures is is all recorded so far. Every class is recorded so that you can find it in the canvas. Okay, and today's class will also be uploaded later when it's ready. Okay, so now from here, then I'll stop here. Let's see this. And goodbye for today. We'll see you on Wednesday. Okay. Okay. Goodbye.